So I've talked a lot about the problems. I'd like to talk about some of the strategies which we're trying to use to solve some of these problems. So we could try introducing new herbicide-tolerant traits into crops. We can do that by genetically modified organisms, traditional breeding, and indeed, of course, the major agrochemical companies are doing that. We could try to reverse the weed resistance in glyphosate, perhaps using a new technology such as RNA interference technology. I'll talk a little bit about that in a minute. And we could try and discover new herbicides which control resistant weeds. So ideally, we should discover compounds with new modes of action, but we could also look for compounds which bind to known targets but which possess completely novel structures which are not subject to the target site or metabolic resistance. But I have to emphasize there's no quick fixes here. And even if we're successful, and I'm sure we will be successful with some of these technologies, these approaches, none of them is going to be a long-term solution. If we continue as we currently are, we're going to have resistance developing to any new product which we bring onto the market. So it is very important that we preserve what we have. So RNA interference technology, that's an interesting new technology being championed by Monsanto. It's actually a natural biological process in which RNA molecules inhibit gene expression. And in 2006, Andrew Fire and Craig Mello, they were awarded the Nobel Prize in medicine for their work on RNAi. Now, Monsanto are hoping to use it because some of the, for example, Palmer amaranth, the resistance comes due to overexpression of the target enzyme EPSP synthase. That's the target for glyphosate. And what they hope is, by topical application of RNA onto the target weeds, that will cause downregulation of the gene, and that will consequently lead to a reduced production of the encoded protein. And that should enable glyphosate to work again. And certainly their early biological testing suggests that this does look very promising as a new technology. However, it's not a technology without problems. It's a RNAi, it's a highly specific molecule which targets one specific gene in one specific organism. RNA is also a very large molecule which is rapidly degraded in organisms. It's expensive to produce and it's very challenging to formulate in a bioavailable form. So there are some challenges which need to be solved there still. What's happened here? Okay. So now I'd like to move on to herbicide research and the challenges which we face in trying to discover new herbicides. So quite often the challenges and the things we're trying to achieve, they're rather contradictory actually. So we want our new herbicides to have very high potency, but we want them to have very low non-target toxicity. We want them to be highly selective to the crops, but we want them to have very broad spectrum control of all of the weeds. We want them to offer prolonged protection and to be stable under all climatic conditions, but we want them to be rapidly degraded in the environment. In addition, we want them to be fast acting. We shouldn't have any cross resistance, ideally a new mode of action, and of course they have to be cost effective. The synthesis has to be cost effective. And that's becoming rather difficult to solve all of these problems because new products are increasingly difficult to find. The easy problems have been solved, so all of the low-lying fruit, if you like, have been picked. New products obviously have to show very significant advantage over older products, and we have increasingly stringent toxicological and environmental safety requirements, which is a good thing. We're all in favor of that, but it does make it more difficult to discover new products. And this is really only achievable in very large multidisciplinary teams. So I'd like to show you what I mean by such a multidisciplinary team. So I'm a chemist, so I'm sitting here, but every day I'm in contact with all of these other disciplines. And we're asking and answering questions like, how active is the compound? Is it selective? What's the biochemical mode of action? How does it behave in the environment? How can it be most effectively applied? How can it be made economically and safely on a large industrial scale? Is it safe for the farmer, to the consumer, to non-target organisms? And in which cultivars and against which weeds is it most effective? So all of these are important questions which need to be solved in the discovery stages of research. So where do we start when we're looking for new herbicides? Well, there's a number of starting points. Very important starting point is the patent literature, so we're looking at what other people are doing, seeing if we can find improvements. But I've mentioned to you that the patent literature is decreasing rather alarmingly. So that's still very important, but less important than it was. We have within Bayer and all the other companies too a very highly developed screening cascade where we're able to screen many thousands, hundreds of thousands actually, of compounds every year. And that's important as a source of new hits. And we also, in addition, this would be in vivo screening, we also do in vitro screening where we take target enzymes, so herbicide target enzymes, and we screen against those in a so-called high-throughput biochemical screening. Then there are a number of other technologies, but one I'd like to just point out at this moment, that's this new technologies based upon protein X-ray crystal structures. And we think that this offers actually a lot of hope for the future, and we are certainly pursuing quite a few research projects along these lines. So I'm trying here to show the research process from the early discovery phase all the way through up here to product launch. So we start with an idea for new chemistry to address obviously a future market. This is often very highly automated, these early stages, so a lot of automated chemistry, a lot of automated screening. We take the hits from that process and we bring them into the optimization where we're trying to establish a very detailed structure-activity relationship. And that's all done using greenhouse testing. The hits from here we take up to the field trial stage where we take the most promising compounds and we test them under real light and soil and climatic conditions. We do the first formulation work. That can have a very big effect, actually, formulation on the activity. And we do the first toxicological tests. Now we're getting close to launch, so the project phase, we're now trying to develop the compound as a new commercial product. Obviously, extensive field trials, very extensive toxicological, ecotoxicological testing, and finally registration. So then the final step is launch. And the important thing about all of this is that it takes 10 plus years. So from here to here, it's 10 years or more in many cases. So there's no, as I say, quick term fixes. So I said I was going to talk a little bit about chemistry, and I'm true to my promise, I shall. So here are the structures of some of the herbicides which have been launched by Bayer. And these structures, I've drawn them in the sort of the slightly abbreviated line form, where every time you see here two lines intersecting, that means there's a carbon atom there, or here, for example. And some of you are probably looking at those structures and you're thinking, they have no real meaning. There's nothing behind those, really. It's not relevant to us. But I want to try and show you that it is very relevant. And I'd like to try and do that, first of all, with the molecule olympocene. That's not a herbicide, but it's an interesting molecule, I think. It was first synthesized in 2012 to celebrate the London Olympics. And the people who synthesized it, they teamed up with an IBM researcher, and they used a new technology called non-contact atomic force microscopy in order to take a picture of olympocene. And this is a photograph, well, not a photograph really, but a picture, an image of a single olympocene molecule. And if you compare this structure here with this image, here we see the 5 rings. So you see also the 5 rings quite clearly in this image. So really these structures, as you see them here, they actually have a real basis in reality. And you might say, well, that's not a very good image. That's a bit blurred. And the reason that it's so blurred is because molecules are so incredibly tiny. So tiny we can't actually imagine how small they are. So the molecules I drew on that previous slide, they were about 1 billionth of a meter long. So that's what we call a nanometer. And that's such a small length that none of us, I certainly can't imagine, and I don't believe that you can either. But if you imagine magnifying a molecule until it's the size of an orange, now we can all relate to an orange. And if you now imagine magnifying that orange by the same factor which you magnify the molecule to get to the orange, you would end up with an orange the size of the Earth. And so I think that's something we can more easily relate to, and that shows just really how tiny molecules are. And yet, despite the fact that they're so tiny, their structure is absolutely crucial to the physical chemical properties and at the end of the day to the biological properties of the compounds which we synthesize and produce. So here's our herbicide pyrosulfatol. Molecular weight, that's a fairly obvious thing we can calculate. But if a chemist looks at this structure, he can see that here we have an acidic OH group, and that's going to be very important to the activity of this compound. I'll come back to that. I can see that we have hydrogen bond donor and 5 hydrogen bond acceptors, and that's very important because this compound has to bind to a protein, and that binding will involve the formation of hydrogen bonds. I can calculate the lipophilicity, so that's a way of working out how fatty a molecule is and the likelihood of it getting through the plant cuticle. We can calculate the polarity, so how likely is it to go through cell membranes. We can look at the volatility. So all of these properties can be calculated just from the structure. And they are properties, as you might expect for a commercial herbicide, which are typical actually for a systemic herbicide, but they haven't been achieved by chance. The point is that we have to target very carefully and very exactly these properties. So let me try and illustrate a little bit about what I mean. So Bayer is well known because we produce, amongst other things, aspirin. Aspirin's pretty good for treating headaches. And you can imagine perhaps your son or your daughter would have a headache, and on the whole, well, one wouldn't think to try and treat that headache by dressing the child up in a raincoat, putting on a nice rain hat, and then we could dissolve the aspirin up in a bucket of water and pour it over them. That's not an effective way of curing a headache. But on the other hand, unfortunately, that is the way which we, of course, apply our herbicides. So we're leaf spraying, at least with post-emergence herbicides. And plants, of course, they're covered in a waxy layer called the cuticle, which is designed to keep compounds out, actually. So when we are formulating our compounds and designing our compounds, it's very important that we consider how we're going to get the compounds into the plant. And once they're in, either through the leaves or the roots, we have to consider very carefully how the compound will behave inside the plant and what sort of mechanisms will be in play in terms of distribution. It's no good just getting the compound into the plant if it doesn't get to the target site where it needs to be. So I'd like to just try and, in more detail, talk about one example. So I'd like to consider the water flow systems within a plant. There's two of them. One is called the xylem. It takes water and nutrients up from the roots to the leaves. And the other one is called the phloem. It takes the products of photosynthesis down to the roots. Now, if you're going to have an effective systemic herbicide, it's very important that you're able to take that compound, it's sprayed onto the leaf, and it has to come down through the phloem to the roots in order that it can come back up through the xylem to the growing tips of the plants. So that's very important. But the trouble is that the flow rate through the xylem going up in this way is 30 to 50 times the flow rate going down through the phloem. So if you rely just on passive distribution between the two, the xylem and the phloem, nearly all of the compound will end up in the xylem going this way, and that's not going to give you an effective herbicide. But luckily, nature's given us a little bit of a help here. So what we see is that the xylem is slightly acidic with a pH of 5.5, whereas the phloem going down is slightly basic with a pH of 7.5. And that's the reason that I said earlier, this acidic hydroxyl group here is very important, because under the conditions of the phloem, the slightly basic conditions, this compound is deprotonated to form this anion, and this leads to the compound being trapped into the phloem. So despite the fact that the flow rate is much less, it becomes trapped into the phloem and goes down to the roots, where it can come back up to control the growing tips. And that's why most modern herbicides actually are acidics. So I guess that's enough of that. So you've been very patient, so thank you very much. The final steps before development, well, we have to obviously check biological efficacy, modes of action. We have to look at environmental fate, behavior, soil behavior. Toxicological studies are very important. New agrochemicals, what you can say is that on the whole, they're very extensively tested, 8 to 10 years. Only 1 out of 140 compounds which we test makes it to the market. And it costs a lot of money. It's an investment of about 260 million Canadian dollars into the future. So that's a big investment, but at the end of the day, it's worthwhile. Here's a new product which we recently launched in Canada called Infinity. It's a mixture actually of pyrosulfatol plus bromoxynil and a safener. It's used for the control of broadleaf weeds in cereals, including kosher. And we add the safener. This compound has actually very good selectivity for cereals, but we add the safener to make sure the selectivity is 100%. Safeners work by increasing the speed at which crops, but not weeds, are able to degrade herbicides. So that's an existing product. What's the outlook for new herbicides? As I've said, increasing weed resistance is a key driver for our current herbicide research programs. And to overcome weed resistance, new herbicides, they need to have either a completely new mode of action, or they need to bind to a target, a known target, but possess completely novel structural types which are able to overcome resistance problems. And one of the recent research programs we've been involved in is the search for new, structurally novel HPPD herbicides. And I'd like to talk a little bit about how we're going about that and the early results. So the question is, how do you find these new structurally novel compounds? Well, one way is to conduct, and I've already mentioned this, high throughput biochemical screening. So where we take the HPPD enzyme and we screen compounds to see whether they're good inhibitors. And we've screened something like 1 million compounds against this enzyme, and we have found actually some interesting hits. Another way is to look at some of these technologies based upon protein X-ray crystal structures, which I mentioned. And one of these techniques is known as fragment-based lead discovery. And we're certainly pursuing this as well with the HPPD enzyme. So we have the HPPD protein crystal structure. That's an important prerequisite. We're able to conduct high concentration screening, which is also very important. And the nice thing about this is that the hits, they tend to be rather small and polar, which is actually features which are very attractive for the development of herbicides. So what do I mean by fragment-based lead discovery? Well, I'm trying to explain that here. So this in a way would be the ideal situation. So here again, this blue box is the enzyme. In this case, it's going to be HPPD. This would be our ideal lead compound. So as before, that would fit very nicely into the enzyme. But the trouble is the chances of finding such a perfect fit are very, very small. Actually, you know, in the high throughput screening, we test perhaps 1 million compounds, and if we discover just a few compounds, maybe a dozen compounds, we're pleased. And many of those perhaps are not ideal for further optimization. And the reason for that is shown here in a way, because a very small change, so we have a very small change in this molecule, and it no longer fits. So even very small variations from the ideal mean that the structure doesn't fit at all, so we don't detect it. We might be very close, but we won't detect it at all. There'll be no biological and no biochemical activity. But if we take a fragment of such a molecule, so here we have a small fragment, that can fit, and the chances of finding such a small fragment-like structure are much, much higher than for a big molecule like this. And that's a sort of a factor, because the thing is, with small fragments, you can cover a much higher proportion of chemical space than you can with a similar size collection of larger molecules. However, what we have to say is that these fragments, they show weak biochemical activity, but probably no biological activity. So there's a lot of work to do to optimize these fragments to get them into proper lead structures like this. This is the sort of thing we've done. And here's an interesting slide. This is one which only a chemist could love. But I want to assure you this is very interesting. I want to try and explain to you why it's interesting. So here we have in the group A and group B very similar molecules. But the interesting thing is that these are fragments which bind to HPPD, whereas these do not bind. And it's interesting to consider, why do these bind, why don't these bind? But the most interesting thing to consider is, okay, these are our lead structures actually, and what can we do to these lead structures to turn them into herbicides? So can we, for example, build lipophilic groups onto these? Can we change the structure in any way to generate new herbicides? And that's what we've been doing, and we've come up actually with some interesting structures. So this is one example, and this is taken from an actual protein X-ray crystal structure of the HPPD enzyme, and this is a new compound which is binding into the active site. It actually binds in a very similar way to pyrosulfatol, so our existing herbicide, but it has a completely different structure. And so we've taken the input from the fragment-based lead finding, we've taken the input from our high-throughput screening, and also from our traditional screening processes, and we've come up with novel structures which are inhibitors of HPPD and which also show good herbicidal activity. But look at the date here. This patent was published on the 31st of March 2011. So if we are successful and this structure we're able to optimize it to a new herbicide product, the earliest we're going to have that product on the market is 2021. So again, no short-term fixes in our research. So let me just summarize. So weed resistance, it's increasing globally, with some weeds now resistant to multiple herbicide modes of action. Weed control management is becoming more complex. The research to find new herbicides is very expensive and it takes a long time. So we said 260 million Canadian dollars, 10 years or more. The market has an urgent need for new herbicides with novel modes of action. However, one has to say that no new major herbicidal mode of action has been commercialized in the past 25 years, and unfortunately none look to be on the immediate horizon. So it's important that we look after our existing herbicidal solutions. We need to protect them by integration with other non-chemical weed control measures. This is my final slide. Saving our herbicides, diversity is the future. This is a very interesting website set up by my Canadian colleagues. I recommend that you all look at it. I'd like to thank my colleagues at Bayer, and most especially I'd like to thank you for your kind attention. Thank you.